Serotaxonomy. Serotaxonomy is a method used to classify organisms based on their serological characteristics, such as the presence of specific proteins or antigens in their blood serum. Procedure of serotaxonomy: collect blood samples. Blood samples are collected from the organisms being studied. The samples are usually taken from the vein of organism and are placed in sterile containers and separate serum. The blood samples are then centrifuged to separate the serum from the other components of the blood, such as red and white blood cells. Then identify antigens. Antigens are identified by performing a variety of tests, such as agglutination tests, complement fixation tests, and ELISA tests. These tests helps to identify the specific antigens present in the serum. Then compare antigens. The antigens identified in step 3 are then compared with those other organisms. This is done to identify similarities and differences in the antigens, which can be used to classify the organisms. Then classify organisms based on similarities and differences in the antigens identified. The organisms are classified into groups. The groups can be based on a variety of characters, such as their serological properties, geographic location, or other physical characteristics. Then validate classification once again. First, collect the blood sample, separate serum, identify antigens, compare antigens, classify organisms, and validate classification. Merits: rapid and cost-effective. Serotaxonomy is a relatively rapid and cost-effective method of classification compared to other traditional methods of taxonomy, such as morphological and molecular taxonomy. Highly specific. Serotaxonomy is highly specific, meaning that it can accurately differentiate between closely related species that may be difficult to distinguish using other methods. Then able to detect cryptic species. Serotaxonomy can also detect cryptic species that may be morphologically similar but have different antigenic properties, helping to uncover hidden biodiversity. Large sample sizes. Serotaxonomy can process large number of sample sizes at once, making it particularly useful in studying large populations of epidemiological studies. Then widely applicable. Serotaxonomy is widely applicable to different groups of organisms, including bacteria, virus, fungi, and some animal species. Stable. Serological characteristics is usually stable, making it possible to store samples for long periods and compare it with samples collected at different times and locations. And complementary. Serotaxonomy can complement other methods of taxonomy, provide additional data which can be used to support or challenge finding from other methods. Then demerits of serotaxonomy. While serotaxonomy has been widely used in the past, it has several demerits, including limited applicability. Serotaxonomy is limited to the study of antigens, which may not always be a reliable indicator of the organism's evolutionary history. In some cases, the serological properties may not correlate well with other characteristics of the organism, make it difficult to draw conclusions about its taxonomy. Then lack of precision. Serological testing is subject to variation and interpretation. Different laboratories may obtain different results, which can make it difficult to compare data across studies. Additionally, the accuracy of serological tests can be affected by factors such as sample preparation, storage, and handling. Limited resolution. Serotaxonomy is typically used to classify organisms at the subspecies or subspecies level, but it may not provide sufficient resolution for higher level classifications. For example, it may be difficult to distinguish between different genera or families using serological data alone. Then limited usefulness for evolutionary studies. Serotaxonomy is primarily a phenotypic approach and does not provide information about the genetic relationships between organisms. As a result, it may not be suitable for studying evolutionary processes such as appreciation that are driven by genetic changes. Limited usefulness for clinical application. While serological tests can be useful for diagnosing infectious diseases, they may not always be reliable. False positive and false negative results are possible, and the interpretation of test results may be influenced by factors such as patient's immune status and aiming of sample collection. Then applications of serotaxonomy. Disease diagnosis. Serotaxonomy can be used to diagnose infectious diseases by identifying the specific antigens or antibodies present in a patient's blood. For example, the ELISA test, enzyme-linked immunosorbent assay, uses serological methods to detect the presence of HIV antibodies in blood samples. Then animal breeding. Serotaxonomy can be used in animal breeding to identify individuals with desirable traits. For example, in cattle breeding, serotaxonomy can be used to identify animals with resistance to certain diseases. Then forensic science. Serotaxonomy can be used in forensic science to identify human remains or to link a suspect to a crime scene. For example, serological testing can be used to determine whether blood stains found in a crime scene match the blood type of a suspect. Evolutionary studies or taxonomy can be used to study the evolutionary relationships between different species. By comparing the serological properties of different organisms, scientists can determine how closely related they are to each other. Then vaccine development. Serotaxonomy can be used in the development of vaccines by identifying the specific antigens that will trigger an immune response. By understanding the serological properties of a pathogen, scientists can design vaccines that will be more effective at preventing infection. Then disease surveillance. Serotaxonomy is also used to monitor the spread and transmission of infectious diseases in populations. By tracking changes in the serological profiles of individuals over time, scientists can identify new outbreaks and determine the effectiveness of vaccination campaigns. Blood transfusion compatibility testing. Serotaxonomy is used to determine the compatibility of donor blood with the recipient's blood type in blood transfusion procedures. Then quality control testing. Serotaxonomy is used to ensure the quality and safety of blood and pharmaceutical products to detecting the presence of harmful substances or contaminants in samples. Once again, let's have a quick recap. Serotaxonomy procedure: collect the blood sample, separate serum, identify antigens, compare antigens, classify organisms. Then merits and validate classification. Merits: rapid and cost effective, highly specific, able to detect cryptic species, large sample sizes, widely applicable, stable, complementary, etc. Demerits: limited applicability, lack of precision, limited resolution, limited usefulness for evolutionary studies, then limited usefulness for clinical applications, disease diagnosis, animal breeding, forensic science, and evolutionary studies, along with vaccine development, disease surveillance, and blood transfusion compatibility testing, and quality control testing. Cytotaxonomy. Cytotaxonomy is a field of taxonomy that uses information about the chromosomes and cellular processes of organisms to classify them. It is based on the idea that the number, structure, and behavior of chromosomes during cell division can provide useful information for identifying and differentiating species. In cytogenetics, the study of chromosomes is used to determine the number and types of chromosomes in a cell, as well as their organization and behavior during cell division. Cytotaxonomy uses information to classify organisms based on their chromosomal characters. For example, they may use differences in chromosome number or presence of unique chromosomal structures or patterns to distinguish between species or to identify new ones. Cytotaxonomy is particularly useful for identifying cryptic or closely related species, while traditional taxonomic methods based on morphology or anatomy may not be sufficient to distinguish between them. It is also useful for studying evolutionary relationship between different groups of organisms, as changes in chromosome number or structure can provide insights into the patterns of speciation and diversification. Procedure: sample collection. Samples of the organism under study are collected from the wild or from culture collections. They may include tissues, cells, or whole organism. Preparation of slides. The samples are then prepared for microscopic analysis. Depending on the type of organisms and the nature of the sample, different techniques may be used for slide preparation. For example, cells may be fixed, stained, and mounted on slides for observation under a microscope. Then microscopic analysis is performed. The prepared slides are then analyzed under a microscope. The cells are observed for their structure, size, shape, and other characteristics. Chromosome numbers, ploidy levels, and other cytogenetic features may also be assessed. Data analysis. The data obtained from the microscopic analysis are then analyzed to determine the taxonomic relationship between different organisms. This may involve comparing the cytological features of different specimens to identify similarities and differences and to group them into taxonomic categories. Taxonomic classification. The taxonomic relationship between different organisms are then used to classify them to appropriate taxonomic groups such as species, genera, families, or order. Which are the procedures? Sample collection, preparation of slides, microscopic analysis, data analysis, and taxonomic classification. Taxonomy classification. The taxonomy classification we just learned. Then advantages of cytotaxonomy: rapid and accurate identification. Cytotaxonomy can be used to identify species quickly and accurately based on their chromosome number and structure. This can be especially useful when traditional morphological and molecular methods of identification are not feasible or reliable. Then phylogenetic relationships. Cytotaxonomy can provide insights into the evolutionary relationship between different species and taxa, as chromosome number and structure can be used as a marker for evolutionary divergence. Then population genetics. Cytotaxonomy can be used to study the genetic diversity and structure of populations within and between species. Chromosome number and structure variations can reveal important information about population dynamics such as polyploidy events, hybridization, and gene flow. In conservation biology, cytotaxonomy can play a role in conservation efforts by providing information on the genetic diversity and uniqueness of populations and species, which can inform conservation priorities and management strategies. Then agricultural and economic importance. Cytotaxonomy can provide information on the genetic variability of crop plants and livestock species, which can be used in breeding programs to improve agricultural productivity and economic value. Then demerits of cytotaxonomy. Limited applicability. Cytotaxonomy is limited in its applicability to certain groups of organisms that can be easily cultured or have accessible cells for study. This means that many species cannot be studied using cytogenetic techniques. Limited applicability. Then technical difficulties. Cytogenetic techniques require specialized skills and equipment and can be technically challenging. Obtaining high quality chromosome preparations can be difficult and interpretation of results can be subjective. Variation within species. Chromosome number and structure can vary widely within a species, making it difficult to draw clear conclusions about the evolutionary relationship between organisms. Then lack of resolution. Cytogenetic studies may not provide enough resolution to resolve complex evolutionary relationships or distinguish between closely related species. Limited information. Cytogenetic studies provide limited information about the biology, ecology, and behavior of organisms. Other types of data, such as molecular and morphological data, may be needed to fully understand the evolutionary relationship between species. Bias towards diploid organization. Cytogenetic techniques are biased towards the study of diploid organisms, which can lead to a incomplete understanding of the evolutionary relationship of polyploid organisms. Once again, let's learn the merits. Quick recap. Limited applicability, technical difficulties, variation with the species, lack of resolution, and limited information, then bias towards diploid organisms. Applications of cytotaxonomy. Some applications of cytotaxonomy include species identification. Cytotaxonomy can be used to identify new species by examining the chromosomes of different organisms. Differences in chromosome number, size, and structure can be used to distinguish between closely related species. Phylogenetic analysis. Cytotaxonomy can help in constructing phylogenetic trees which show the evolutionary relationship between different species. By comparing the chromosome number and structure of different organisms, scientists can determine how closely related they are and how they evolve over time. Then conservation biology. Cytotaxonomy can be used to assess the genetic diversity of different populations and determine whether they are in the risk of extinction. It can also help in identifying species that are not yet recognized and need to be protected. Then crop improvement. Cytotaxonomy is useful in crop breeding programs as it helps in the identifying plants with desirable traits. By examining the chromosomes of different plants, breeders can determine which ones are most closely related and which ones have the genetic characteristics they are looking for. In medical research, cytotaxonomy can be used to study the genetic basis of diseases and identify new therapies. By examining the chromosomes of patients with different diseases, scientists can determine which genes are involved in and develop treatment that target those genes. Then plant breeding. Cytotaxonomy is important in plant breeding to produce new plant variants with the desirable traits. Chromosome manipulation and hybridization techniques can be used to create new plant varieties with improved disease resistance, yield, and quality. Once again, applications: species identification, phylogenetic analysis, conservation biology, crop improvement, medical research, plant breeding. Some other trends. Integrative taxonomy. Integrative taxonomy is an approach that combines multiple lines of evidence, including morphology, genetics, and ecology, to identify undesirable species. This approach aims to provide a more comprehensive understanding of species diversity and to improve The accuracy of taxonomic classification. DNA barcoding. DNA barcoding is a technique that uses a short standardized DNA sequence to identify species. This approach can be used to quickly and accurately identify species from a wide range of organisms, including plants, animals, and fungi. Then, phylogenomics. Phylogenomics is a field that combines molecular data with traditional morphological and ecological data to reconstruct an evolutionary relationship among organisms. This approach can help to resolve taxonomic uncertainty and to provide a more accurate understanding of the relationship among species. Open access database. Open access database such as Global Biodiversity Information Facility or GBIF provide a platform for sharing taxonomic data and making it more accessible to researchers and the general public. It can help improve accuracy and efficiency of taxonomic research. Citizen science. Citizen science involves the participation of non-experts in scientific research, including taxonomic research. This approach can help to increase the amount of data available to taxonomists and to engage the public in the process of understanding and protecting biodiversity. These newer trends in taxonomy are helping to improve our understanding of the diversity of life on Earth and to develop more accurate and efficient methods of classifying organisms. Next topic: ethics in taxonomic publications, authorship, correspondence, and suppression of data. Ethics in taxonomic publication. For having the honesty in scientific research, the principles of ethics is needed for all the researchers and students. Like ethics in taxonomic publications is also very important, and here the assignment is discussing absent the ethics about the ethics in taxonomic publications, authorship of taxonomic paper, correspondence, and suppression of data. An author of taxonomic paper can criticize work of others in a dignified and courteous manner. The author should not make an attempt to attack the other person's personality. Mistakes of others can be pointed out or rectified in a constructive manner. An author of a taxonomic publication should follow the ethics of taxonomic publication. A taxonomist may often want to correspond with other taxonomists to collect information for his publication, while requesting these kinds to help of research should be specific and should not ask in a general form like provide all these materials you have. It's not like that. And moreover, while requesting, one should avoid including anything private which the otherwise does not like it to be read. A good taxonomist will not deliberately suppress any relevant information that publishing his findings. However, this sometimes happen in ignorance, and through that, and occasionally suppression is done deliberately to protect one's identity, privacy, and personal information. So, like this, authorship of taxonomic paper important one. Authorship is based on the curriculum provided by each author who had their significant contributions to the study. So, correspondence. A taxonomist often finds it essential to correspond. A taxonomist often find it essential to correspond with other taxonomists requesting for information on types, reprint of publication, and reference of taxa. So they will be requesting for information on types, reprint of publication, and reference on taxa. While requesting reprint, one has to be specific and should not ask for general terms. If he wants several reprints of the author, he has to explain briefly in this lecture how far he has done his work and what is his future plans. He should also include copies of his publication along with the request. And when one receives the reprint, it should be acknowledged. While requesting reprint, the title of the paper, journal or which it was published, and the year should be written. Then request for loan or subscription should be specific. And it should also state that he will take utmost care in handling the specimens while studying. And the specimen should be returned as early as possible. Then while writing letters, one should avoid including anything private. In recent times, most scientists prefer to use electronic mails, so prefer that method. if the other person is comfortable with it. Then suppression of data. Data suppression refers to the process by which suppression of data is happening. So most public repeats of this. No, this is not a good practice. So a scientist may give details of his findings to other taxonomists, and the works or taxonomist may fail to mention that the information is based on the findings given by the author or scientist. We should mention that sometimes the individual may suppress data deliberately to protect the identities, privacy, and personal information of others. That's okay. Then data suppression is used when there is a chance that the information contained is publicly available. Report should be used to reveal or infer the identity of specific individuals. Then it should be protected. And it is important to properly analyze the data to know why the information is being withheld. This is especially important. Then authorship. An author of scientific research manuscript is considered markedly and spiritually to the scientific content. The need to credit an author. Credit of authorship is important in academic, social, and financial implications. Authorship should be based on contribution provided by each author who has made significant scientific contribution to the study. Guidelines for authorship. Who is not an author? According to scientists, who is not an author? Acquisition of funds, supervision of fund work, minor laboratory support, administrator support. People who just provide these are not authors. Types of authors, there are different types. First author is someone who conceptualize the study and contribute every aspect of the research process, study, design, all this. Then co-authors, those who significantly contribute to the research work and in preparing of the manuscript. Then correspondence author, one who takes responsibility for the manuscript doing its submission. Then unethical authorship practices, grant authorship, gift authorship, ghost authorship. Now let's learn about the next topic, punctuated equilibrium theory and gradualism. Gradualism is a universal theory that has been applied to many other sciences aside from biology. Gradualism has been applied to geography. Gradualism has been applied to linguistics or language. Gradualism has even been applied to the political science. Here we are going to learn about gradualism and evolution. So, the gradualism model of speciation holds that small changes accumulate to form big changes. The punctuated equilibrium model suggests that rates of changes accelerate over short periods in small peripheral populations and then stabilize for long periods in large central populations. So, gradualism is a universal theory that has been applied to many other scientists. We just learned this, I'm sorry. Gradualism continued. Put plainly, gradualism is the theory that change occurs over time. To use large words, gradualism is a theory proposed by a man named James Hutton in 1795 that states profound changes is the product of accumulative change. It is more often than not contrasted with catastrophism. The gradualism model of speciation holds that small changes accumulate to form big changes. The punctuated equilibrium model suggests that rates of change accelerate over short periods in small peripheral populations and then stabilize for long periods in larger central populations. Evolutionary theory and gradualism. Charles Darwin, the father of evolution, was identified by Charles Lyell's book on the subject, influenced by Charles Lyell's book on this subject. He even incorporated it into his famous theory. Charles had first read Lyell's first book, Principles of Geology. Gradualism today. Gradualism is often confused with phyletic gradualism, which is a biology specific term. Phyletic gradualism is a term that was created by two scientists to contrast with their own model. The Model of evolution created by Aldrin and Gould is called the punctuated equilibrium. Example of gradualism. One good example should be the turtles found on the Galapagos Islands. They changed slowly fit into their respective environments. Morphology, low explanations. Gradualism model, morphological change, punctuated equilibrium model. Punctuated equilibrium. The punctuated equilibrium is a way to explain how species change. It is very similar to the tree-like diagram but seems more modern. The straight section is when a species is in stasis. When the line splits, the species become two separate species. Punctuated equilibrium occurs because genetic variations are passed down quicker, thus increasing the rate of evolution and speciation. Then Tyrannosaurid fossils and mollusks in the Turkana Basin are examples of punctuated equilibrium. Differences between the punctuated equilibrium and gradualism. The differences between the two are very obvious. Gradualism says that it is cumulative change that changes a species. The punctuated equilibrium shows that changes occur quickly and in short periods of time. Both refer to evolution of species over time. However, gradualism states that evolution occurs slowly over a long period of time. Punctuated equilibrium occurs when there are periods of apparent stasis interrupted by sudden change. Gradualism, punctuated equilibrium. Gradualism, morphology. This represents the tree of life under Darwin's gradual theory of evolution, where one species turns into another species through slow, smooth, gradual changes over long periods of geological time. Then punctuated equilibrium, an evolutionary tree under a punctuated equilibrium model. Most change in morphology, like macroevolutionary change, takes place over geographically shorter time periods in small populations, such that translational forms are unlikely to be fossilized. Gradualism versus punctuated equilibrium. Gradualism, species descended from a common ancestor and gradually diverge in morphology, acquiring unique adaptations until they are reproductively isolated. Punctuated equilibrium, long periods of apparent stasis punctuated by brief periods of sudden and rapid change. Fossil record confirms the punctuated equilibrium model, also eliminates the necessity of finding missing links. Next topic, neutral theory of molecular evolution. Molecular evolution. Molecular evolution is the process of change in the sequence composition of cellular molecules such as DNA, RNA, and proteins across generations. The field of molecular evolution uses principles of evolutionary biology and population genetics to explain patterns in these changes. Neutral theory of molecular evolution. The theory was introduced by Japanese biologist Moto Kimura in 1968. The theory suggests that most evolutionary changes at the molecular level are due to random genetic drift rather than natural selection. According to this theory, the majority of mutations that occur in DNA sequences do not have any effect on organisms' fitness. Most mutations that occur in DNA sequences are neutral or nearly neutral, meaning they do not significantly affect an organism's fitness. The rate of molecular evolution is determined primarily by the rate of mutation, not natural selection. Natural mutations can become fixed in populations through a process called genetic drift, which is the modern fluctuation of allele frequencies over time. Natural selection can still act on molecular evolution, but it primarily affects the rare mutations that are beneficial or harmful to an organism's fitness. The theory predicts that number of differences between the two species or populations should be proportional to the amount of time since they diverged from a common ancestor. This prediction has been supported by studies comparing the DNA sequences of different species and populations. The neutral theory also predicts that vast majority of molecular changes should be silent, meaning they do not result in changes to the amino acid sequence of a protein. The neutral theory of molecular evolution changes the traditional view that natural selection is the primary drive of evolutionary change at the molecular level. The neutral theory does not deny the existence of natural selection, but rather suggests that it may a smaller role in molecular evolution than previously thought. The neutral theory has been a controversial topic in evolutionary biology, with some researchers supporting it as a valid explanation for the patterns of genetic diversity that we observe in nature, while Others argue that natural selection plays a more significant role in shaping genetic variation. Advantages of natural selection, natural neutral theory of molecular evolution. Explaining the high levels of genetic diversity, the neutral theory predicts that the majority genetic variations is due to the accumulation of neutral mutations which do not affect the fitness of the organism. This hypothesis has been supported by empirical data. Once again, explaining the high levels of genetic variation. Diversity. The neutral theory predicts that the majority of genetic variations is due to accumulation of neutral mutations which do not affect the fitness of an organism. This hypothesis has been supported by empirical data, including the high levels of polymorphism observed in many populations. Predicting the rate of molecular evolution, the neutral theory predicts that the rate of molecular evolution is proportional to the underlying mutation rate and population size. This Advantages of neutral theory of molecular evolution. Explaining the high levels of genetic diversity, the neutral theory predicts that the majority genetic variations is due to the accumulation of neutral mutations which do not affect the fitness of an organism. This hypothesis has been supported by empirical data, including the high levels of polymorphism observed in many populations. Predicting the rate of molecular evolution, the neutral theory predicts that the rate of molecular evolution is proportional to the underlying mutation rate and population size. This prediction has been supported by empirical data from a wide range of experiments and organisms, including bacteria, virus, and eukaryotes. Simplifying assumptions. The neutral theory is based on a simple set of assumptions which make it easier to test and compare with other assumptions and evolutionary hypotheses. The theory assumes that mutations occur randomly, that they have no effect on fitness, and the genetic drift is the primary force of driving evolution. Disadvantages of neutral theory of molecular evolution. Overestimation of the role of genetic drift. The neutral theory assumes that genetic drift is the primary force driving molecular evolution, while natural selection is relatively unimportant. However, recent research has shown that natural selection is often a major driver of molecular evolution. Lack of evidence for truly neutral mutations. It is difficult to determine whether a particular mutation is truly neutral, meaning it has no effect on an organism's fitness. Therefore, a neutral theory may overestimate the frequency of truly neutral mutations. And limited applicability of complex traits. The neutral theory is most applicable to molecular traits such as DNA sequences or protein structure and may not be as useful for understanding complex traits such as behavior or morphology. Molecular drive. Molecular drive is a term coined once again, this last point. Limited applicability to complex traits. The neutral theory is most applicable to molecular traits such as DNA sequence or protein structure and may not be as useful for understanding complex traits such as behavior or morphology. Molecular drive. Molecular drive is a term coined by Gabriel Dober in 1982 to describe evolutionary processes that change the genetic composition of a population through DNA turnover mechanisms. Molecular drive is a term used to describe the process by which certain genetic elements such as transposons, so selfish gene elements, increase the frequency within a population over time. These elements are called selfish because they do not necessarily confer a benefit or direct benefit to the organism carrying them, rather promote their own replication and spread within the genome. Molecular drive can occur through various mechanisms such as unequal crossing over during meiosis, preferential segregation during gamete formation, or biased gene conversion. Then examples of molecular drive include transposable elements, homing endonucleases, and meiotic drives. Molecular drive is an evolutionary process like natural selection and neutral drift that changes the genetic composition of a population through generations. It is distinct from natural selection and neutral drift in that it emerges from the activities of a number of universal mechanisms for DNA turnover such as gene conversion, unequal crossing over, slippage, transposition, and retrotransposition. Gene conversion, it occurs when two related but divergent sequences exist in the same cell and can be substrates for recombination. It occurs when two related but divergent sequences exist in the same cell and can be substrates for recombination. The outcome of gene conversion is a unidirectional transfer of genetic sequence. The outcome of gene conversion is a unidirectional transfer of genetic sequence information from a donor sequence into a highly similar recipient sequence. Gene conversion occurs when two related but divergent sequences exist in the same cell and can be substrates for recombination. The outcomes of gene conversion is unidirectional transfer of genetic sequence information from a donor sequence into a highly similar recipient sequence. And unequal crossing over. Improper pairing between homologous chromosomes. Homologous chromosomes are made up of chromosomes pairs of approximately same length, centromere position and staining pattern for genes with the same corresponding loci. One homologous chromosome is inherited from the organism's mother, the other is inherited from the organism's father, is called unequal crossing. Then the result is a crossover chromatid with one copy of the segment and another with the 3 copies. The replication slippage. It is the misalignment of DNA strands during the replication of repeated DNA sequences and can lead to genetic rearrangements. Then transposition, the phenomenon of moving genetic segments from one location to the other in genome is known as transposition. Retrotransposons, they are the type of genetic components that copy and paste themselves into different genomic locations for converting RNA back to DNA through the process reverse transcription using RNA transcription intermediate. Molecular divergence. Molecular divergence refers to the accumulation of genetic differences or changes in the DNA sequence or protein structure of molecules or organisms over time. It is a concept commonly used in evolutionary biology and genetics to understand the relationship and evolutionary history of different organisms. And molecular divergence occurs due to accumulation of genetic mutations in DNA sequence of organisms. These mutations can arise by various mechanisms such as DNA replication, errors, environmental factors, or exposure to mutagens. Which are the processes? DNA replication error, environmental factor, or exposure to mutagens. Over successive generations, these mutations become fixed in the population, leading to genetic differences between individuals and ultimately between species. Molecular divergence can occur at different rates in different ages and can be influenced by various factors including the species, population size, generation time. Molecular divergence is highly relevant in the field of evolutionary biology and genetics. Understanding evolutionary relationship, molecular divergence allows scientists to reconstruct the evolutionary relationship between different species. By comparing DNA or protein sequences, researchers can determine the degree of genetic divergence between organisms, providing valuable information about their common ancestors. When estimating divergence time, by analyzing the molecular divergence between species, scientists can estimate the time since their last common ancestor. Then species identification and taxonomy. Molecular divergence plays a crucial role in species identification and classification. Then medical and pharmaceutical applications. Molecular divergence analysis has practical implications in medicine and pharmaceutical research. Mechanisms and factors influencing molecular divergence. Mechanisms and factors influencing molecular divergence can be grouped into two main categories: genetic mutations, external factors, genetic mutations, point mutations. These are single base changes in the DNA sequence. They can occur through substitution, where one nucleotide is replaced by another. Then insertion and deletion. These mutations involve the insertion or deletion of nucleotides in the DNA sequence. And duplications. These mutations result in the duplication of a DNA segment, leading to an increased genetic material that can undergo further mutation. That is duplication. Then rearrangements. Large scale structural changes such as inversions, translocations, chromosomal rearrangements can contribute to molecular divergence. Then external forces, environmental factors, environmental conditions such as temperature, pH, or exposure to mutagens like radiation, chemicals can increase the rate of genetic mutation and contribute to molecular divergence. Examples of molecular divergence: speciation and genetic divergence. Darwin's finches. The Galapagos finches studied by Charles Darwin exhibit remarkable molecular divergence. Different species of finches have evolved quite unique beak shapes and sizes which correspond to their specialized feeding habits and ecological niches. Then adaptive radiation. Hawaiian honey creepers. The Hawaiian honey creepers are a group of birds that have undergone adaptive radiation in the isolated Hawaiian Islands. They exhibit diverse beak sizes and sizes specialized for various food sources such as nectar, fruit, and insects. Molecular divergence analysis has revealed genetic variations in beak-related genes contributing to their adaptive radiation. And morphological cloak and phylogenetic relationships. HIV evolution. In human immunodeficiency virus exhibit molecular divergence due to its high mutation rate and rapid replication. Then I'm analyzing the genetic sequences of HIV strains from different individuals and geographic regions helps track the virus's evolutionary history, understand its transmission pattern, and develop effective treatments. Then advantages of molecular divergence. Phylogenetic relationships. Molecular divergence data can be used to reconstruct evolutionary relationship among different species or populations. Genetic variation. Molecular divergence provides insight into the genetic variation present within and between populations. It allows researchers to identify genetic differences and variations in DNA sequences. Then species identification. DNA seemed based molecular divergence analysis can aid in species identification. By comparing genetic sequence, scientists can differentiate between closely related species that might appear similar extremely but have extinct genetic signatures. Molecular clock. Molecular divergence data can be used to estimate the timing of evolutionary events and measure the rate of genetic change. By analyzing the accumulated genetic differences between species or populations, researchers can develop molecular clocks. Disease studies. Molecular divergence studies are valuable in understanding the genetic basis of diseases. Next topic, molecular clock. The molecular clock is a method that uses biomolecular data to estimate the amount of time needed for a certain amount of evolutionary change to occur. The molecular clock was first proposed by Mercer Clinton and Linus Pauling in the 1960s, and since then it has become a widely used tool in evolutionary biology. This is done by analyzing the biomolecular data, such as the number of changes or substitutions in nucleotide sequence of DNA and RNA or the amino acid sequence of proteins. Substitution is a type of mutation where one nucleotide is replaced by another. Substitution is a type of mutation where one nucleotide is replaced by another. Assuming that the nucleotide or amino acid sequence mutate at a constant rate, the number of substitutions over time is equivalent to the evolutionary rate. The molecular clock is also known as gene clock or evolutionary clock. The molecular clock hypothesis argues that DNA and protein sequences mutate at a constant rate over time and among different organisms. Mutations may be harmful, beneficial, or neutral. Harmful mutations have a negative impact on an organism's evolutionary fitness or its ability to survive and reproduce. On the contrary, beneficial mutations have a positive impact on an organism's evolutionary fitness. Most mutations are neutral. They have no effect on the organism's evolutionary fitness. Molecular clock can be used to determine when different species last shared a common ancestor, to put evolutionary events in chronological order, both of which are essential to the construction of phylogenetic trees. Molecular clocks are also useful for studying species that do not fossilize well. For example, using this analysis, researchers found that animals and fungi last shared a common ancestor more than a billion years ago. Molecular clocks are useful to study species that do not fossilize well, and also useful in creating phylogenetic trees. Then limitations. RNA, DNA, or protein sequence may change at irregular bursts instead of at a constant rate. Some DNA, RNA, or protein sequences may appear to change at a smooth average rate, but actually have some deviations from that average rate. As a result of natural selection, some genetic changes are favored over others. The same DNA, RNA, or protein sequence substitutions may be occurring at different rates in different organisms. Some DNA, RNA, or protein sequence evolve faster than others. Next, human mitochondrial molecular clock. The human mitochondrial molecular clock is the rate at which mutations have been accumulated in the mitochondrial genome of human during the course of human evolution. A major goal of scientists is to develop an accurate human mitochondrial molecular model, which could then be used to confidently date events that occurred during the course of human evolution. The two main methods of estimation is phylogeny-based methods and pedigree-based methods. Pedigree methods estimate the mutation rate by comparing the empty DNA sequence of a sample of parent or offspring pairs. The number of new mutations in the sample is counted and divided by the total number of parent-to-child DNA transmission events to arrive at a mutation rate. Then phylogeny-based methods are estimated by first reconstructing the holotype of the most recent common ancestor, or MRCA, most recent common ancestor of a sample of two or more genetic lineages. The human mutation rate is usually estimated by comparing the sequence of modern humans and chimpanzees and then reconstructing the ancestral halotype of the chimpanzee-human common ancestor. Then, according to the paleontological record, the last common ancestor of humans may have lived around 6 million years ago. It only provides information on the maternal line of descent and cannot provide information on the paternal line or on genetic variation in the nuclear genome. Genetic equidistance. Genetic equidistance is a concept that the genetic distance between two or more sequences is proportional to the time since they diverged from a common ancestor. The genetic distance between two or more sequences is proportional to the time since they diverged from a common ancestor. If two sequences have diverged for the same amount of time, they should be equally different from each other, regardless of the direction of nature of the evolutionary changes that have occurred during that time. The concept of genetic equidistance is closely related to the molecular clock hypothesis, which proposes that rate of genetic evolution is approximately constant over time. If this hypothesis holds true, then the genetic distance between two sequences should be proportional to the time since they diverged. Genetic equidistance has been observed in a wide range of organisms and genes, including mitochondrial DNA, ribosomal RNA, and certain protein coding genes. It has been used to estimate the divergence times to various species and populations, and then to infer the populations and mechanisms of molecular evolution. Then limitations: genetic equidistance may be affected by other factors such as genetic drift, selection, and recombination. Advantages: genetic equidistance remains an important concept of molecular evolution and phylogenetics. It has provided valuable insights into the patterns and processes of genetic diversity and evolution across the tree of life. Then homologous sequence of protein and DNA. Homologous sequences are sequences of DNA or protein that share a common evolutionary origin. They are sequences that have descended from a common ancestral sequence. It is subjected to divergent evolution over time. Homologous DNA sequences are similar in their nucleotide composition and arrangement. Homologous protein sequences are similar in their amino acid composition and arrangement. Homologous DNA sequences encode homologous protein sequence and vice versa. And homologous sequence can be identified by comparing the sequence of interest to other sequences in a database, also by using computational methods like pairwise sequence alignment, multiple sequence alignment, or phylogenetic analysis. This method can reveal regions of similarity and a difference between sequences and can be used to infer evolution relationship among sequences. Importance: homologous sequences are important in molecular biology and genetics as they provide insights into the structure, function, and evolution of genes and proteins. Homologous DNA sequences can be used to infer evolutionary relationship among species, to identify functional elements such as regulatory regions and protein coding genes. Overall, homologous sequences provide a powerful tool for understanding the relationships and diversity of life at the molecular level.